Statement 1 says, in the Lac Operon, the Z gene codes for beta galactosidase. Correct. Which is responsible for the hydrolysis of lactose into galactose and glucose. Correct. So statement 1 is true. In addition to lactose, glucose or galactose can also induce Lac Operon. This is incorrect. Only lactose can induce Lac Operon and hence the name. So this statement is false. So right answer is option number 1. So you take around 45 seconds to attempt such questions. You read the statements carefully and then only mark your answers. So let's try to understand this particular question. Z gene or Z gene codes for beta galactosidase. Lactose is a disaccharide. Disaccharide means it has two monosaccharide which are linked together by glycosidic linkages. So the two monosaccharides are galactose and glucose. The link between galactose and glucose is beta 1 4. Therefore, the enzyme that is able to cleave this bond is called beta galactosidase. And Z gene or Z gene codes for this particular enzyme in Lac Operon. So this gene is called structural gene because it codes for structure of the enzyme. So everything is correct in this particular statement. What is wrong in this statement? Glucose and galactose can also induce Lac Operon. See, the whole purpose of switching on Lac Operon is to metabolize lactose. And the product of lactose metabolism is glucose and galactose. So once all the lactose is consumed, obviously you will shut down Lac Operon because the enzymes can only act on lactose. So this diagram explains why glucose and galactose cannot serve as inducer of Lac Operon, because only lactose can bind to the repressor, and once the repressor is bound by lactose, it cannot bind to operator. And now there is nothing blocking the RNA polymerase. So RNA polymerase will continue and perform the transcription process. So this is called polycistronic gene because all the three important structural genes are transcribed together. They are back to back in tandem. So see, a variety of questions can be asked based on this topic. So one question can be, what are the structural genes? So these three are the structural genes. And the most common question is, what does Z code for, or Y codes for, or A codes for? The answer is Z codes for beta galactosidase, Y codes permease enzyme, and A for transacetylase. Now another question which they can ask is, which enzyme is always present in low levels? So the answer is permease. Because this permease is important, then only if this is the bacteria and in the medium lactose is present, you need a mechanism for this lactose to enter in the cell. So permease does that function and it allows lactose to enter in. Once lactose enters in, it binds to the repressor and then in large amount the permease enzyme will be produced. So it can then enter in bulk amount. Another question asked on this topic was, what is upstream and downstream of the structural enzyme? So upstream is the initiation site and downstream is the termination site for transcription. And what else? The I stands for inhibitor, not inducer. And what is inhibitor? Inhibitor is the protein, or it is called repressor, which binds to the operator. So you can notice operator is downstream of promoter. Okay, so transcription will happen in this direction. So this is called downstream of promoter. If anything is here, then it will be called upstream of promoter. So that's all about this topic. Thank you so much for watching my video.